The microenvironment is crucial for chronic lymphocytic leukemia, in particular the tissue microenvironment. But what we usually do in the lab is using cells isolated from the peripheral blood of the patients, because of course it's the place where we have most access. So this could be a problem, because the cells that are in the tissue, they are in contact with other cell types. They are in an adhesion context, let's say, that is very different from the fluidic that they can find in the peripheral blood. So that's why I realized that currently we need to develop new models that will allow us to mimic the tissue setting in vitro. Of course, this is quite difficult because you need to add many cell types to the culture system, and this is not really possible in conventional 2D culture. You can just, let's say, go up to 2 cell types together. So that's why I think that we need to develop new models that will allow us to increase the complexity. Of course, we have animal models to study also what happens in the tissue, but they have their limitations, and we are not really in a human context. So what I'm proposing now, and not only me, now it's a very hot topic in the field of cancer, because many therapies failed. Probably this means that we are not really studying the disease in the proper context. So we are now trying to establish new models by using, in my case, for instance, scaffold-based systems where we co-culture in a scaffold cells of leukemia and cells of the microenvironment, and then we also use dynamic growth in bioreactor to add even an additional level of complexity. In this system, for instance, we were able to study the phenotype of the cells inside the scaffold, so in a tissue-like context, and outside of the scaffold, so in a more peripheral blood-like context. And we found that they have a different phenotype, and we validated these results also in vivo in the patients. And last, also very important, because for instance the new target therapies such as BTK inhibitors, they work by mobilizing the cells from the tissue into the peripheral blood, where they lose the protective effect of the microenvironment and they eventually undergo apoptosis. So now this is not possible to study these mechanisms of mobilization into the culture, of course, but this is possible in 3D culture, where you have a tissue setting and the cells can be mobilized in the bioreactor.